We're going to talk a little bit about mitosis today. We've talked about asexual reproduction and the fact that it uses a process called mitosis. Some of you remember maybe talking about it in middle school, but let's refresh our memories what it means. So mitosis is a type of cell division used for three main things: asexual reproduction, as we discussed earlier, growth, maybe adding new cells to a multicellular organism, or repair. Multicellular organisms can use mitosis to repair dead cells that need to be replaced. So just because we are a multicellular organism that normally reproduces by sexual reproduction, it doesn't mean that we don't use mitosis in our bodies. We use mitosis not for reproduction, but we use it for growth. As you're growing, teenage years, younger years, children, adolescents, you're adding new cells to your body to grow larger, and you're also repairing dead cells that get worn out over time. So during mitosis, there's a couple of things that happen, but the main thing that we think about is that the DNA, or genetic information, is going to replicate, which means to copy, and the cell is going to split in half. It's going to divide up organelles and divide up the DNA into two daughter cells, they're called. Not son cells, but daughter cells. These daughter cells are identical to the parent and identical to each other. So they're almost kind of, if you would think of it as clones, but these are natural clones. With that said, there is no genetic variation. They're identical to each other. So we're going to talk about some of the problems that can come along with having no genetic variation, unlike with sexually reproducing organisms that have a lot of variety. DNA, remember, is our hereditary material, allowing us to pass on our traits to our offspring, and it's organized into structures called chromosomes, which contain our DNA. Our normal chromosome number in our normal body cells is referred to as 2N. N refers to the number of different types of chromosomes we have, and 2 essentially means that we have 2 copies of each type. So in the case of humans, we have 23 different types of chromosomes, and we have 2 of each kind in our normal body cells. 1 came from our mother, 1 came from our father, for a total of 46. We often refer to this number as our diploid number. Di, again, refers to 2. So we have 2 copies of each. 1 set came from our mother, 1 set came from our father. During mitosis, that diploid number would be maintained. You wouldn't split the number of chromosomes in half. You would still have a total of 2N, or the diploid number of chromosomes. So how does mitosis really happen? I know this looks a little bit extensive, so you're going to probably need to pause in order to be able to draw each of the phases. Some of you may remember the phases from middle school. You've done this before. We're going to refresh our memories because we're going to be talking about these phases again and revisiting them and comparing them when we talk about sexual reproduction and meiosis. So let's look at each of the phases. When it comes to mitosis, we would have a normal parent cell, and these structures inside the nucleus represent our chromosomes. And in this case, I have 2 of them colored red and I have 2 of them colored blue. And that represents the 2 that came from the original mother cell, if we're talking about an organism that reproduces sexually, and 2 that maybe came from the father cell, that same thing, from an organism that reproduced sexually. In the case of an organism that completely reproduces asexually, we remember there's no real mother or father, but they still have 2 copies of each kind of chromosome. So our 2N number in this case would be 4. We have a total of 4 chromosomes. Remember, in humans, we have a total of 46 chromosomes. Our first phase, which really isn't truly a phase of mitosis, is called interphase. During this phase, we're really carrying out life processes, but if the cell is ready to go through cell division or mitosis and divide, the DNA is going to replicate. It's going to make a complete copy of itself, and it's going to prepare for mitosis, reproducing organelles and things like that. The DNA at this point really is formed in something called chromatin. You don't see it as these nice little X-like structures of chromosomes yet, but for visual purposes, we're going to show them this way. So now you can see that we made copies of our DNA. They now look like almost like little X's with each other. We have an original and copy. So it almost appears like we have 8, but we have 2 copies of each one. Our real first phase of cell division is called prophase. During prophase, If you're an animal cell, the centrioles become visible, and they produce these things called the spindle fibers that the chromosomes attach to, and the nuclear membrane is going to disappear. During the next phase, those chromosomes are going to line up in the center, attached to the spindle fibers. The centrioles are going to go towards the poles, stretching out those spindle fibers. And we're going to see that they don't necessarily arrange themselves in a specific order. It's relatively random. But they line up in the middle so that during the next phase, anaphase, they can pull apart from each other evenly so that in the end, each new cell gets one of each type of chromosome. In the center, when they're lined up during metaphase, they're attached to the spindle fibers, and the chromosomes are still attached to each other, the original and its copy, and they're attached by a structure called a centromere. During anaphase, that original and its copy split at the centromere. The original goes one way, the copy goes the other way of the chromosome, and we eventually have them starting to go towards the poles or opposite ends. Our next phase, telophase, we see that if we're an animal cell, we start pinching in this cell membrane. We see that the chromosomes are towards each end, and we can see the beginnings of a nuclear membrane reforming. At the end, we divide up the cytoplasm in a process called cytokinesis, and we divide up the organelles. The nuclear membrane completely reforms, and you can see that we have a restored chromosome number of 4. We have 2 copies of each of those chromosomes, so we have a total number of 4 chromosomes in each cell, identical to the parent, identical to each other. When we talk about mitosis, we talk about the cell cycle a little bit. It's just the cycle of phases and events that a cell goes through in its life. Most of the time, cells are in a phase called interphase. It's used to be called a resting phase. It really isn't actually resting then. It's just not actively dividing. It carries out all of its life processes during this phase of interphase. Most cells, for the majority of their lifespan, are in interphase. During cell division, where it's going through prophase, metaphase, anaphase, telophase, and cytokinesis, the active parts of mitosis, it's a very short period of time for most cells. A lot of cells can carry out this process within about a half an hour. I'd like you to try to think of a way to try to remember the phases of mitosis. Some of you guys might have remembered the IPMAT, I-P-M-A-T, taking the first letter of each of the phases. But remember, interphase technically isn't a phase that we talk about during mitosis. It's kind of not included, even though we need to discuss it. So maybe you can come up with your own way to remember the order of each of the phases, and maybe you want to include cytokinesis for the division of the cytoplasm. One of the issues with the cell cycle, and we'll talk about this a little bit more in detail in class, is that if this cell cycle isn't regulated well and gets out of control, and the cells just keep dividing and no longer have control of division, we end up with a process of cancer. We'll talk about the control of this and how genetics can come into play during cancer. But just understand that when the cell cycle loses control and regulation, that's technically considered cancer. And that's it for mitosis. What I'd like you to do tonight is I'd like you to just briefly study each of the phases and make sure that you're familiar with what happens in each phase.